Tuendelee na biochemistry, na kama ambavyo kwenye video iliyopita tulivyoona kuhusu classification of enzymes, na kwenye video hii tunaendelea kuhusu the rate of enzymes controlled reaction pamoja na factor affecting the enzymes enzyme controlled reaction. Kwa usisahau kusubscribe channel, usisahau kushare the video, na pia nikwambia channel hii sasa vitakuwa vinaendelea kuhusu vipindi vya physics, advanced mathematics, BAM pamoja na chemistry. Kwa hiyo tuanze. The rate of enzyme controlled reaction. The rate of an enzyme reaction is measured by the amount of substance changed or amount of product formed during a period of time. So this is chemistry, kwamba rate ya reaction tunapima kwa amount ya reactant iliyotumika, yani substrate au product. Now the rate is determined by measuring the slope of the tangent to the curve in the initial stage of the reaction is shown in the figure below. So figure yenyewe ni hapo kwamba unakuwa na tangent ile pale, unaangalia slope, baada hapo slope inakupa rate ya reaction. The steepness of the slope, the greater the rate. If activity is measured over a period of time, the rate of reaction usually falls, most commonly as a result of fall in substrate concentration, as will be discussed later. Kwa rate ya enzyme controlled reaction inakuwa kubwa pale ambapo tunakuwa tuna substrate nyingi. Substrate ikipungua, most of times the rate of enzyme controlled reaction huwa nayo inapungua. So now let's jump to the factors affecting the enzyme's activity. Factors affecting the enzyme's activity. When investigating the effect of a given factor on the rate of enzyme controlled reaction, all other factors should be kept constant and at optimal level whenever possible. Initial rates only should be measured as explained above. Kwa hiyo unapima initial rate na pia unaziweka other factor to be constant. So the activity of an enzyme is affected by general environmental factors such as temperature and pH, and also by particular chemicals that specifically influence that enzyme. Kwa hiyo factor ya kwanza kabisa ni substrate concentration. Substrate concentration. Tunachokisema ni kwamba for a given amount of enzyme concentration, the rate of an enzyme reaction increases with an increasing substrate concentration. Kwa rate ya enzyme controlled reaction inaongezeka na pale unavyozidi kuincrease substrate concentration, up to a point where any further increase in substrate concentration produces no significant change in the reaction rate. Kwa kuna point ambayo ukiongeza substrate haitabadilisha chochote kwenye reaction. Why so? Kwa nini? This is because kwenye hiyo point all active sites of the enzyme will be occupied with the substrate. Kwa hapo utaweza kufanya chochote kwa sababu active sites zote zimekuwa occupied. Kwa substrate nyingine zinazokuja zinatakiwa zisubiri kwanza product iondoke kwenye active site, ndipo ambapo zenyewe ziweze kuingia. Kwa graph yetu ambayo inaonyesha how enzymes are affected by the substrate concentration ni hapa, na huku ndiko ambako tumefikia saturation point. The increase in substrate concentration furthermore will flatten the graph because all of the active sites, they are occupied by substrate. Our second factor is enzyme concentration. The rate of enzyme controlled reaction is directly proportional to the enzyme concentration as long as no other factor are limiting the rate. Kwa hiyo rate ya enzyme controlled reaction inazidi kuongezeka kwa kadri unavyoongeza enzyme, lakini kuna wakati ambao unafika graph inakuwa flat ghafla, kama ambavyo unaweza kuona hapa, graph inakuwa flat ghafla. Kwa nini? Kwa sababu substrate inakuwa imeshaisha. Kwa if substrate concentration is limiting, graph itakuwa flat kabla. Lakini kama substrate concentration iko ya kutosha, is excess, basi graph yako itakuwa straight kama hivyo. Factor 3 ni temperature. Factor 3 ni temperature. Heating increases the molecular motion. Thus, the molecules of the substrate and enzyme move more quickly, and chances of their bumping into each other are increased. As a result, there is a greater probability of reaction occurring. The temperature that is promotes maximum activity is referred to as optimum temperature. Hii factor temperature imeelezewa vizuri sana kwenye kitabu cha Understanding Biology, lakini wakati naandaa hizo notes sikuwa na Understanding Biology, so I used the only BS. I advise you to go and study the temperature factor in advanced biology. So if the temperature is increased above this level, then the decrease in the rate of reaction occur despite the increase in the frequency of collision. Kwamba unavyozidi ku increase temperature from the optimal level, the rate of the enzyme controlled reaction decreases. Why so? This is because the secondary and tertiary structure of the enzyme have been disrupted and the enzyme is said to be denatured. So most mammalian enzymes have a temperature optimum at about 37 to 40 degrees centigrade. If the temperature is reduced to near or below freezing point, enzymes are inactivated, not denatured. Kwa unavyopunguza temperature, enzymes zinakuwa inactivated, lakini unavyo increase temperature zinakuwa denatured. So they will gain their catalytic characteristics influence when higher temperature are restored. Na kama ambavyo unaona hapo kwamba unajua freezing, tunavyoweka vyakula kwenye friji vinakuwa viharibiki kwa sababu kwenye ile temperature bacteria wanashindwa kuishi, na pia enzymes zinazoweza kukatalyze breakdown ya hizo nyama zinakuwa zinashindwa kufanya kazi. So the freezing is the most recent food preservation technique. The method inactivates the digestive enzyme of the particular food substance, thus making it impossible for them to decompose food. Also, freezing prevents growth and multiplication of microorganisms. So as you can see here, this graph shows the rate of chemical reaction increase in temperature to optimum. At optimum here, the temperature seems to be 37, and then if the temperature is increased from optimum, then the rate of chemical reaction tend to fall. In temperature here, we have one of the factor here is called temperature coefficient, or Q10. Temperature coefficient, or Q10, this is the change in the rate of reaction for each 10 degrees centigrade rise in temperature. The effect of temperature on the rate of reaction can be expressed as temperature coefficient, Q10. So Q10 is the rate of reaction at x plus 10 degrees centigrade. Then rate of reaction at x degrees centigrade. So increase in the rate of reaction by the increase of 10 degrees centigrade, then divided by the normal price that the one who is increasing the rate of reaction is doing that reaction. Kwa unaangalia reaction speed yake kawaida na speed baada ya kuongeza 10 centigrade, then kuchukua divided by unapata hiyo temperature coefficient. So over a range of 0 to 40 degrees centigrade, the rate of enzyme controlled reaction double for every rise of 10 degrees centigrade rise. Thus it has the Q10 of 2. Lakini pia the last factor is pH of the medium. Na pH tunasema ni negative logarithm of the hydrogen ion concentration. Tenda kuisoma hiyo kwenye acid base and salts kwenye chemistry. So the precise three-dimensional molecular shape, which is vital for the functioning of enzymes, is partly to the functioning of hydrogen bonding. These bonds may be broken by concentration of hydrogen ions. By breaking the hydrogen bonds, many changes in pH can effectively denature enzymes. Kwa hiyo unapokuwa unavunja hydrogen bonds kwenye enzymes yako au anywhere, hiyo inasababisha the change in pH. Na change in pH mwisho siku inaweza kusababisha kudenature enzymes, kama ambavyo tulisoma factors for protein denaturation, and we said proteins they are enzymes. So the optimum pH is that at which the maximum rate of reaction occur when the pH is altered above or below this value, the rate of enzyme activity diminishes. Kwa hiyo kuna optimal pH, na kama ambavyo tunaweza kwamba kuchange pH tunakuwa na change hydrogen ion concentration. Kama ambavyo tunaweza kuona kwenye hii table, tuna different enzymes and they have different pH for their function. But this also is the graph which shows how the pH affect the rate of enzyme controlled reaction. So in this video, let's end up here. In the next video, we will discuss about the inhibitors, and then we shall jump to the allosteric enzymes, and then we shall jump to cofactors and things like that. So stay with me, subscribe to the channel, share the link to your fellows. Na video nyingine za hesabu level advance zitaendelea kurushwa kwenye channel hii. Kwa kaeni hapa, kaeni tu hapa, kaeni subscribe channel, share kwa marafiki zako, tuweze kuendelea kushiriki pamoja. Niwatakia wakati mwema na masomo.